Well, hello everyone. In the next 3 or 4 videos, we are going to review last year biology exam, that's 2023 paper 2 questions, and suggest answers to them. After going through each video, do not hesitate to post questions and comments, and then do not forget to subscribe to get the next videos in the series. When we are done with the videos, we may also look at the practical for last year and then come up with some predictions and guidelines that may help you to prepare for 2024. Right, so just stick around. All right, so let's get into it. Muddy our hands, make our hands dirty. Okay, so this is WAEC Biology 2023 Paper 2. It is divided into 2 sections, Section A and B. So Section A, you have 4 questions and you have to answer 2. Then Section B, you have just 1 question, which is compulsory. You answer everything. So it's set out in such a way that it cuts across most of the topics from Form 1 to Form 3 or first year to third year. So in this particular video, we're just going to look at the first 2 questions in Section A. Okay, but before that, let's look at some of the do's and don'ts. Okay, so the first is avoid using words like help, right, and then aids, right, helps or help, and then aid or aids. Okay, you just go straight forward. Wherever you have a situation where you have to use something like help, just use for, right? So the hand is used for writing, not helps for in writing. Okay, so just straightforward. Then for adaptation questions, structure and function is required. Okay, it's not high grade to just put function there alone. You'll not get anything. And structure too, you'll not get anything for that. You have to connect the structures to the appropriate functions. Okay, then the spelling must be correct to score. So the spelling must also be correct to score, right? In especially where the word makes all the difference, you know, in the statement that you make. You know, it's about photosynthesis. Okay, plants use chlorophyll for photosynthesis. Photosynthesis becomes very important word in that statement, so you need to spell it correctly. Okay, then where we have taxonomical names or classification names, you also need to usually begin with them with capital letters. You will not be able to italize them since you are not using computers and all that. So you just go with starting with the capital letter. Then that will be fine. So let's look at the first question. So the first question was explain briefly the following terms. So 1 is taxonomy. It is a branch of biology which involves a classification, you can say systematic, that's okay, of living things into groups based on their similar characteristics, genetic, structural, or cellular organization. You can see that these are all strokes. So you can just end at characteristics and you'll get the mark for that. Okay, then we go to II, which is binomial nomenclature. Binomial nomenclature. This is the scientific method of naming organisms by giving the organism 2 names, which are derived from the genus and species. Right, now you can see that it at certain points in the answers there are semicolons, right? So that means that the examiner will award you a mark for that. Question like this will go for 2 marks. So if you say any of these, give any 2 here, right? You have to get your the full mark, which is 2 in this case. We go on to B. Say 2 characteristics each of members of the following classes of organisms. We have I. So we have the hepatici, right? There's the following classes of organisms. We have the hepatici. So the hepatici, what are some of their characteristics? What are the characteristics of organisms that are in this class? So we have gametophyte. Gametophyte is dominant, right? The part that bears or produces the gamete is a dominant one. It is dorsoventrally flattened, right? Thallus shows dichotomous branching. Thallus refers to the body, right? So that some part of the body, it divides into 2. That's what we refer to as dichotomous, right? So dividing into 2 and usually flat on the floor, on the ground, wherever it will be. Then you have capsule split into 4 halves when ripening to release spores. Capsule bears the sporangium or produces the sporangium. Alright, so we go to the next one. Capsule possesses oval or spherical shape. Okay, then we have plant possesses leafy lobes or thalloid. Okay, or thalloid, still referring to thallus. So for hepatici, they tend to have a non-differentiated body, right? The body is not distinct into the various organs of the plant or even the systems of the plant. That's the shoot, the roots, and then the leaves and all that are not distinct. Okay, so we say that they do not have true root, stems, and leaves, and they are also non-vascular. So that's the hepatici. Then we go to the class Muci. So if class Muci, you have the first one, just like hepatici. You also have gametophyte, which is also the dominant generation. You know that they have 2 generations in plants. You have the sporophyte generation, then the gametophyte generation. So the gametophyte in Muci said that it's differentiated into stem-like, leaf-like, or root-like structures. And then the leaves are apetiolate. That is, the leaves do not possess stalk. Okay, we also say that it's sessile. That's another term for apetiolate, sessile, right? Which means the leaves are directly attached to the stem-like, part of the plant. Then we have haploid or dominant gametophyte, as I've already said, just like in the hepatici, so that you see the similarity between them. Spirally arranged leaves. Then we have septate rhizoids. They're multicellular. They are multicellular organisms. Then let's go to question C. Sub-question C under question 1. So I'll give one example each of the members of the following classes of organisms. You have I, hepatici. So I have liverwort, Marchantia, Riccia, Pelia. So Marchantia, Riccia, Pelia are all scientific names. So start with capital letter. Don't have to italize it, but we just write them with capital letters. Then we have Muci. You have moss. Then Brachymenium and Funaria. They all follow the same trend as the Marchantia and Riccia in hepatici. Then we come to D. State 4 characteristics of each of the members of the following classes of organisms. We have Chilopoda. So with Chilopoda, say that the animals here, they are arthropods, right? So the arthropods that have segmental bodies. Bodies are elongated. They are flattened bodies. They are nocturnal, mostly active at night. They possess a pair of simple eyes, possess a pair of antennae, possess a pair of mandibles or maxillae, possess a pair of long legs on each abdominal segment. We can just say segment. Okay. Right, so let's go to, right, so they are terrestrial animals. They are mostly predatory or carnivorous, and they possess a pair of poisonous claws of it should be venomous claws. Okay, good. Gaseous exchange is by trachea. So they have the tracheal system through which air passes through, and then gaseous exchange will occur internally. They are diplopoda or diplopoda. Here, bodies of the members or the animals here are divided into two parts, head and trunk. They possess two pair of legs on each body segment. They are terrestrial animals. They are scavengers. Pointed anterior end. The anterior end refers to where they have the mouth parts, the pair of antennae, so pair of mandibles. Possess a cylindrical body. They possess a pair of antennae. Antennae are quite short here. Then you have E. Give one example of each of the members of the following classes of organisms. They have Chilopoda, so centipede. Then I Diplopoda, millipede. All right, so that ends question 1. Okay, the sum total of marks here is 20 marks. Okay, usually 1 mark for just a one-word answer. And if you have a sentence, you also go for 1 or 2. If it's a definition, usually 2 marks. If adaptation, structure, and function, each one is 1. So you get 2 for each statement you write under adaptation, as you see in question 2. So question 2, here we go. A. State for adaptations of the small intestine in humans to its functions. Here. So here we have the structure or the structures in bolded, right, bold-faced, so that you can easily see them, and then the functions are not bolded. Okay, so always go with the structure and function. The examiner will just be following that way. You can, of course, talk about function and then the structure can follow, but then, I mean, it's not well, that's fine. It's up to you. Okay, the inner walls have many folds. That's the small intestine there. Okay, so for maximum absorption of substances. There another structure here, have increased or large surface area for maximum absorption of substances. That one too is okay. Then are longer, talking about the length, to increase time for maximum absorption. So the longer the food stays in the small intestine, the longer then it allows time for absorption diffusion to occur, and then as much of the end products that are useful then get into the bloodstream. Then we have finger-like projections of villi which facilitate diffusion, absorption of food for easy or fast diffusion. Then we have here richly supplied with blood capillaries or vascularized. Okay, so just go for each one. You can say vascularized for quick transport of digested food into the bloodstream. You can even say vascularized for faster diffusion. So that you can see that in all of this, we are not using help, we are not using aid. Just go straight to it. Then we have B. Question 2b. Name 3 elements that are supplied to plants through the application of fertilizer: nitrogen, potassium, phosphorus. So these are the elements. Please don't give the symbols, right? Don't give the symbols. Don't give the symbols. We want the elements, not the atoms or whatever. Don't give the symbols. So the spelling is important, especially in potassium for some of us. So you note it. Then still 3 ways by which water is important to plants. So you have medium for translocation of food, raw materials for photosynthesis, needed to maintain cell turgidity, medium for enzymatic or metabolic activities, medium for transport of gametes in lower plants, right? We know of the mosses and ferns, liverworts, so right. So that's why mostly they have or part of the reason why they are found in moist environment. Transpiration of water allows cooling effects on plants. Good. Then we move on to D. Just go back. D is here the question. Let me drop D to the next page. All right. Okay. So we have explained briefly the reason why a high concentration of salt in the soil tends to reduce transpiration in plants. So here we are correlating high concentration of salt in water to the fact that then plants will tend to release less water in the form of water vapor through their stomata. So let's look at it. The cell sap of root hairs is more dilute than the soil water. So get it. We have a situation whereby around the root that's in the soil, there's so much salt. Salt here is referring to iron. So it could be someone maybe who has over fertilized, if I should use that word. Probably think about abusing of the soil. Something like that. Has not followed the required regimen for application of fertilizer and has over fertilized. The soil is going to have so much ions. That's what referred to as salt. Here is going to create a concentration gradient, but this time not favorable to the plant. Why? Because you are going to have the cells, the root hairs, having more water than the soil. So what is going to happen? By osmosis, the water will move from the soil, the plants, the roots, to the soil, right? Which is not advantageous to the plant at all. Okay, so the next point, say causing water to move out of the cell sap into the soil. Then less water will reach the leaves. Then the reduction in transpiration. So if less water now flowing through the plants, then it means that the plants are going to have a reduction in transpiration, because more water is rather leaving the plant root into the soil. Instead of water moving into the plant or into the roots, we have water rather leaving the roots into the soil. So reduction will be highly reduced. Exposure to high concentration of salt adversely affects growth of plants and wilting occurs. Okay. Then we also have, the last one. Name 3 proteins secreted by the stomach and the pancreas. When you talk about proteins, we are talking about enzymes that break down proteins into smaller polypeptides and peptides. Okay, so usually most of these enzymes in the protein or that digest protein or breaks down protein chemically are in their inactive form. I would like you to, tell me why, most of the proteins are, most of the proteins are secreted in their inactive form, right? You can put it in the comments below, or you can share with me anyhow, maybe on the WhatsApp platform or whatever, so that you get to know why are enzymes that digest proteins are released in their inactive form, right? So examples of proteases are pepsin, that is released as a form in pepsinogen, and is activated by HCl. Renin usually is in that form because it's not so much that Well, I want you to find out about that. I will not give you the clue. So renin is more in young mammals or young mammals that serves to coagulate milk proteins so that pepsin can break them down. Then we have trypsin, chymotrypsin. Then we have carboxypeptidase. So the trypsin, chymotrypsin, and carboxypeptidase as released or secreted by the pancreas, also in their inactive form. For trypsin, we have trypsinogen, we have chymotrypsin is secreted in the chymotrypsinogen as chymotrypsinogen in the, that's the inactive form. Then carboxypeptidase, we have the procarboxypeptidase. So all these are inactive forms and they are all activated. Trypsinogen is activated by enterokinase, right? An enzyme in the small intestine, usually around the duodenal areas of the small intestine, the duodenal areas of the small intestine. Then once the trypsinogen has been activated in trypsin, it goes to now activate chymotrypsinogen into chymotrypsin, right? All these are enzymes that chemically digest proteins into peptides and polypeptides and peptides, and then finally we have amino acids. Carboxypeptidase secreted in the secreted as procarboxypeptidase is now activated by trypsin into carboxypeptidase, and then they go ahead to digest proteins as earlier stated. Okay, so this is the first two questions of the exam 2023. So as we have said, there will be 3 or 4 more of these videos so that we can do justice to the rest of the questions. Stay with us so that at the end of the last video we'll look at some of the, as we go on, we'll look at some of the things that you need to pay attention to as we prepare for the 2024 biology, right? Okay, so that's it. That's all we have for this video. So check out the next one. Okay, see you soon.